Hello once again, Bio 220 students. Today we'll do Chapter 5, Microbial Metabolism. Now, metabolism is essentially the buildup and breakdown of nutrients within a cell. And these are just the variety of chemical equations necessary to provide energy and create substances that continues life. There are catabolic pathways which break down large molecules into simple components and release energy in the process. There are also anabolic pathways that build up large molecules by combining simpler components, and these use up energy. In other words, catabolic and anaerobic pathways are linked by the transfer of energy, where catabolic reactions provide the energy needed to do anabolic reactions. Although microbial metabolism can cause disease and food spoilage, many pathways are actually beneficial rather than pathogenic. Now, the pharmaceutical industry uses a variety of bacteria and fungi to actually produce a lot of antibiotics, such as penicillin, which is derived from a fungus. There's also bacitracin, erythromycin, and other treatments such as vaccines, vitamin supplements, or vitamins, and enzymes are also produced or derived from microbial metabolism. Enzymes help speed up metabolic reactions. ATP, the energy molecule, is used by microbes at our cells to manage energy needs. Catabolic reactions will couple with ATP production, producing more energy molecules, and anabolic reactions will couple with ATP breakdown, the using of energy. Now, again, catabolism, that's the part of metabolism where large molecules are broken down into smaller components, and this provides energy and building blocks for anabolism. It's considered exergonic, which means it produces available energy or releases energy. There is also anabolism, which uses energy, which takes in energy, ergo it is endergonic, and is used to actually take building blocks to form larger molecules or to store molecules, like forming glucagon from glucose sugar. Essentially, catabolic processes release energy, such as in the case of glycolysis, breakdown of glucose, or there is anabolism, which uses up energy, such in the case of taking small amino acids to form long chains into proteins. And this just involves a cycle of going from adenosine diphosphate and inorganic phosphate into a combined adenosine triphosphate molecule. The breakdown of that third phosphate group in ATP is a bond that stores energy, but when broken down, the energy is released. Metabolic pathways are sequences of enzymatically catalyzed chemical reactions in a cell. The metabolic pathways are determined by enzymes, and these enzymes are actually originally encoded by our genes. Now, there is collision theory, which states that chemical reactions occur when atoms, ions, and molecules simply collide. The activation energy is simply how much energy is needed in the collision for the chemical reaction to actually continue from reactants to products. And the reaction rate is the frequency of collisions, how often they occur, or how often they have enough collision energy to occur. Now, reaction rate, or the speed of reaction, is increased by enzymes, or by increasing often the temperature, pressure, or concentration of what's being reacted. Catalysts, again, they speed up chemical reactions without being altered themselves. And enzymes are biological catalysts. Essentially, they're catalysts that we make ourselves, and enzymes will act on a specific molecule, a specific substrate. And this substrate enzyme complex involves a lowering of the activation energy or the energy barrier needed to move that reaction forward. This is just a diagram showing how, in one case, a reaction occurs without a catalyst and it has to overcome a large energy barrier before it can go from reactants to products, whereas a catalyst might help keep two components A and B in one place, which lowers the collision energy needed to get them to react. And by lowering the energy barrier, it speeds up the reaction. Now, substrate makes contact with the enzyme's active site, the location where it will actually bind to the enzyme and form the complex. This substrate is then transformed and rearranged into the end material or products, which are released from the enzyme, and then the enzyme is essentially unchanged and can react with more substrate, converting them to products. This just shows that there is a specific site on enzymes where substrates can actually bind to and then be converted into products, either by splitting them up or combining them. But the enzyme does not get used up or destroyed in the process. Thank goodness. Now, enzymes have a specificity for particular substrates, and the process of turnover number is the number of substrates a molecule, the number of substrate an enzyme can convert to product per second. It can be easily from 1 to 10,000 molecules of substrate per second. These are very effective. As far as enzyme naming, biochemists have made a very consistent naming structure for most enzymes. Sort of like how biologists have named many bacteria based off of scientific naming, biochemists have a very good naming system for enzymes as well. They're typically end with the word ACE if they're an enzyme, and they're grouped based on the type of reaction that they speed up. There are oxidoreductases, which speed up oxidation reduction reactions. There are transferases, which transfer functional groups from one molecule to another. There are hydrolases, which help break down molecules with the addition of water. And there are lyases, which remove atoms without water. There are isomerases, which help rearrange the internal bonding and arrangement of atoms. And ligases, which help join molecules such as ATP together. Enzyme components involve an apoenzyme, which is the protein portion. The string of amino acids involves a cofactor, which is not a protein component, but a type of cofactor could be a coenzyme, which is an organic cofactor. Essentially, these assist in the enzyme working. And then there are holoenzymes, which are apoenzyme, which is the combination of an apoenzyme, protein structure, and the cofactor, the thing that helps the protein enzyme. So this is just showing how the combination of an apoenzyme and a cofactor to form the holoenzyme is what is actually a functional enzyme that can act on a substrate. Now, there are those that assist enzymes, such as electron carriers. There's nicotinamide adenine dinucleotide, NAD+. Also, something more common in plants, nicotinamide adenine dinucleotide phosphate, NADP+. These are basically different types of cofactors, flavin adenine dinucleotide, FAD, and coenzyme A, things that we use as animals. There are things that help or could hinder enzyme activity, which is temperature, pH, the concentration of substrate, as well as competitive inhibitors. These all affect how good or how poorly an enzyme does its job. High temperatures or extreme pH ranges can actually denature or unfold the protein chains an enzyme has made, thus making it no longer functional. In the case a concentration of substrate is high, the enzyme will likely catalyze at its maximum rate, whereas if the substrate is low, the enzyme will catalyze at its more lower, at less optimal rate. Now, this is just showing on the left a active protein, whereas on the right, either a extreme pH or temperature has caused it to unfold and be denatured, which means it is no longer functional. And this just shows enzyme activity, where an enzyme may become more active with temperature and then quickly drop off in activity and usefulness if the temperature gets far past its denaturing point. And then a similar curve can be shown in pH. This actually emphasizes that enzymes typically have a very specific pH range that they'll work. There are enzymes that work in our stomachs, which need a very acidic pH. There are enzymes that work in our intestines, which need a more basic pH. So this just shows that enzyme activity, it's very specific to certain conditions, temperature, pH, and a variety of other things. And enzyme activity will increase as substrate concentration increases, but there is a maximum where at which the enzyme will no longer be able to pump out. It will be at its maximum production, essentially. There are things that interfere with enzymes called competitive inhibitors, which essentially they'll jump in line and latch onto the active site of an enzyme and basically compete for the substrate. Because the active site is no longer available, the substrate can't actually be worked on and turned into products. There is the normal binding of a substrate to the active site, and sometimes a competitor will bind to part of the active site, preventing the whole substrate from latching on. Now, two example molecules are sulfanilamide and PABA, which can be inhibitors. Usually inhibitors have a component on them that's pretty similar to a certain part of the actual substrate, which is why they're able to bind a part of the enzyme. There are non-competitive inhibitors. Essentially, they interact with another part of the enzyme, which is called the allosteric site. So instead of the active site, some other part of the enzyme. And this is called allosteric inhibition. So essentially the inhibitor binds to another part of the site and contorts the protein or the enzyme in a way that the substrate can no longer bind because the active site has been altered. Now feedback inhibition, essentially the thing that is made in the end, the product that is made after the substrate is worked on, sometimes if it gets into a high concentration, it can act as an allosteric inhibitor, causing the enzyme to no longer produce more of that product, just to help prevent an excess in that product. This essentially helps preventing continuously making too much of something. So, when the product of an enzyme substrate reaction acts as an allosteric inhibitor, it basically, once too much of the end product is made, it will contort the enzyme to where it can no longer bind the substrate. There are ribozymes, which consist of RNA that functions as a catalyst by cutting and splicing RNA, other chains of RNA. Now, in oxidation reduction reactions, there are two simultaneous processes. One is the oxidation, or the removal of electrons from a source, and then the reduction, where something is gaining more negative electrons. In order to occur, oxidation reduction must occur at the same time, which is why they're overall called a redox reaction. where an oxidation reaction is paired with a reduction reaction, where electrons are removed during the oxidation reaction and added back in during the reduction reaction from one substance to another. So, one substance that has an electron is oxidized by transferring it to another substance, and the substance that gains the electron is what is reduced. In biological systems, electrons and protons are removed at the same time. This is an equivalent to a hydrogen atom. In other words, a hydrogen atom is just a proton and an electron. So removing both effectively means the transfer of a hydrogen atom. Biological oxidations often involve or are often dehydrogenations, or the removal of hydrogen. Case in point, where a proton and electron are both removed to a coenzyme, thus causing the original molecule to become reduced, or I should say oxidized because it loses an electron, and the cofactor becomes, or coenzyme becomes, reduced because it gains the electron. The point of moving a proton, or I should say a proton along with an electron, is because moving a negative charge or a positive charge on its own can be difficult, but if you move a positive and negative charge at the same time, you're effectively moving a neutral area of space. Therefore, coupling protons and electrons together, it allows this process to occur more easily. Now, ATP is generated by a process called phosphorylation, which is essentially adenosine diphosphate has a phosphate group added onto it. And this requires inputting energy in order to form that bond between inorganic phosphate and ADP. So it consumes energy when you're going from ADP to ATP. Now, ATP that is generated when high energy phosphate is added to ATP, well, essentially that's how you generate ATP, through substrate level phosphorylation. This basically just shows that there is a bond connecting inorganic phosphate on one molecule, and it is eventually transferred onto ADP to form adenosine triphosphate. During oxidative phosphorylation, electrons are transferred from one electron carrier to another along a long chain of electron transport systems. Essentially, an electron moves from one component to another, and on the way, this helps within the membrane. This helps release energy that's needed to generate ATP from adenosine diphosphate. And this is essentially the very eloquent process where an electron flows from one molecule to the next, allowing energy to transfer and form ATP, where the final source of that electron is going to be oxygen, which is combined with two hydrogen protons to form water. The process of phosphorylation, it occurs in light-trapping photosynthetic cells, so things where photosynthesis happens. Essentially, the light energy is converted, or it's used to form ATP by the transfer of electrons, oxidation, from chlorophyll to pass through a system of carrier molecules, that long chain that we showed before, or are showing right now, I should say. Now, there are a series of enzymatically catalyzed chemical reactions in metabolic pathways, which are used for energy production. And essentially, this extracts energy from organic compounds and stores it in the chemical ATP, where ATP is a more easily transported molecule that our bodies are better at breaking apart to get that energy in that chemical bond. Carbohydrate catabolism. It's the breakdown of large carbohydrates into smaller components, and during the process releasing energy by breaking bonds between those small components. Examples include glycolysis, which is the breakdown of glucose to form energy, molecules of ATP. There is also the Krebs cycle, and there is the electron transport chain system, which was displayed earlier. Respiration is just simply processes that can occur in the presence of oxygen as the final electron receiver during that electron transport chain, or when oxygen is low, there is fermentation. Oxidation of glucose to pyruvic acid helps produce ATP and the coenzyme NADH. Now in the preparatory stage, basically what happens during glycolysis or the breakdown of glucose, 2 ATP are used up initially. So glycolysis does involve inputting a little bit of energy at first. But then eventually glucose is split to form 2 molecules of glycerolaldehyde, 3-phosphate. And then that molecule is further converted and broken down along the way. And even though it took some initial ATP at the start to do glycolysis, eventually more ATP is produced in the end. Now, the energy conserving stage, this basically is where 2 molecules of glyceraldehyde 3-phosphate are oxidized to 2 pyruvic acid molecules. 2 ATP are produced in this stage, as well as 2 NADH coenzymes. And this is basically a long way of showing how you start out with glucose, sugar, and 2 ATPs, and eventually it forms 2 molecules of pyruvic acid, 4 molecules of ATP, 2 coenzymes of NADH, as well as 2 available protons. It's an overall net gain of 2 molecules of ATP for each molecule of glucose that is used up. Now, additional pathways for glycolysis. This involves pentose phosphate pathway, which uses pentose sugars, so 5 carbon sugars, and produces NADPH. It operates simultaneously with glycolysis. And then there is the Etner-Dordorff pathway, which produces NADPH and ATP. It does not involve glycolysis, and it will occur in bacterium like Pseudomonas, Rhizobium, and Agrobacterium. Cellular respiration, which is essentially oxidation of molecules that liberates electrons to then go down the electron transport chain. The final electron acceptor comes from the outside of the cell and is usually some inorganic substance. ATP is generated by this oxidative phosphorylation. Krebs cycle is involved in aerobic respiration. Essentially, pyruvic acid that was produced by glycolysis is further oxidized and decarboxylation occurs, essentially the removal of a CO2 molecule. This results in 2-carbon compound that attaches to coenzyme A, forming active acetyl-CoA or acetyl-coenzyme A and NADH. Now, the Krebs cycle essentially involves the oxidation of acetyl-CoA, acetyl-coenzyme A, to produce coenzyme NADH, FADH2, and ATP, as well as liberates a carbon dioxide molecule as waste at the end. This is effectively how we breathe out carbon dioxide. Oxygen is needed to be absorbed or breathed in to be as a final electron acceptor during the electron transport pathway, but during the process, carbon dioxide is produced as waste. Typically, you'll go more into these cycles in cellular and molecular biology if you take that course. Now, aerobic respiration involves electron transport chain. This occurs in the plasma membrane of prokaryotes as well as the inner mitochondrial membrane of eukaryotes. It's a series of carrier molecules such as flavoproteins, cytochromes, and ubiquinones, where electrons move from one carrier molecule to the next, are slowly oxidized and reduced as electrons pass down the chain, and energy is released during this process and is used to produce ATP by chemoosmosis, or the transport of chemicals. Case in point was shown here. Aerobic respiration continued. It involves chemoosmosis, where electrons form NADH, and they pass down the electron transport chain while protons are pumped across the membrane. This pushes proteins from an area of low concentration to an area of higher concentration, thus going against the gradient, which helps form a proton motive force later on, where protons can then transfer back in and combine with that electron with oxygen to form water. Essentially, protons in higher concentration on one side of the membrane diffuse through a membrane protein called ATP synthase, which the transport of that proton then causes ATP to form. So essentially, the purpose of the electron transport chain is it helps protons move across the membrane to an area of higher concentration. Therefore, in that area of higher concentration, they can move back in through ATP synthase, which uses the energy that is produced to form a bond between ADP and inorganic phosphate to form ATP. So in reality, this is the involvement of the movement of electrons and protons. Now, in aerobic respiration, the final receiver or the final acceptor of that electron is going to be a molecule of molecular oxygen, O2, which is why we need to breathe it in to allow these processes to occur. In reality, this aerobic respiration, which is the full breakdown of glucose, in the end uses up 38 ADP and 38 inorganic phosphates to produce 38 ATP energy molecules. So it produces carbon dioxide and water, which we'll breathe out as we exhale. But it shows that one glucose molecule and just a little bit of oxygen can produce an incredible amount of energy molecules. In carbohydrate catabolism, each NADH can be oxidized in the electron transport chain to produce 3 molecules of ATP, and then each FADH2 coenzyme can produce 2 molecules of ATP as they get oxidized. So essentially forming NADH and FADH2 during glycolysis, the breakdown of glucose, allows these 2 molecules to be used up later where they will become oxidized and form ATP, even more than just the simple breakdown of glucose. While the final, in anaerobic respiration where oxygen is not present, the final acceptor of the electron is going to be something else besides oxygen. Anaerobic respiration actually produces far less energy than aerobic respiration, which is why it's not favorable. It's preferable to have oxygen. Now, aerobic respiration, there are electron acceptors such as nitrate, sulfate, and carbonate ions, and the products of which are going to form nitrite, molecular nitrogen, water, or in the case of a sulfate acceptor, you form hydrogen sulfate and water. In the case of a carbonate electron acceptor, you'll form methane and water. This is just a chart of many examples of ATP yield during prokaryotic anaerobic respiration of one glucose molecule. And effectively, in the end, basically, Aerobic respiration produces a lot more ATP than simple anaerobic respiration without glycolysis or the Krebs cycle. Now, the process of fermentation, this releases some energy from oxidation of organic molecules, and it does not require oxygen. It also does not use the Krebs cycle or the electron transport chain, cycles that really help produce a lot of ATP. So it uses an organic molecule as the final electron acceptor, and because it skips the Krebs cycle and electron transport chain, it only produces a very minimal amount of ATP. And for large organisms like us, anaerobic respiration isn't worth it. But it is good enough for bacteria and lower and single-celled organisms and simple-celled organisms. Essentially, glycolysis occurs where glucose is broken down to pyruvic acid, and, well, if we do produce anaerobic respiration in our muscles, we can use pyruvic acid and degrade it into a little bit of ATP to help extend our muscles' activity. But this is what causes lactic acid to form. In homolactic fermentation, this produces lactic acid only, and in heterolactic fermentation, lactic acid and other compounds are also produced. Usually these are not good for our bodies. Glucose is oxidized to pyruvic acid, but it's not further broken down, such in the case of aerobic respiration, but instead, glucose is oxidized to pyruvic acid, and then the process produces NADH. But in alcohol fermentation, which produces ethanol and carbon dioxide, glucose is oxidized to pyruvic acid, and then pyruvic acid is converted to acetaldehyde and carbon dioxide. NADH reduces the acetaldehyde to ethanol. So essentially, ethanol, which is not a good component inside, say, our muscles, or useful, it's usually a toxin in most cells that go through anaerobic fermentation or use alcohol fermentation, such as yeast. So types of fermentation. There's glycolysis, which eventually provides pyruvic acid material, and each of those pyruvic acid that are produced, they could go through lactic acid fermentation, which produces 2 molecules of lactic acid, or go through alcoholic fermentation, which produces 2 molecules of ethanol. Essentially, the end result of pyruvic acid from glucose, it depends on the organism, but there is a wide variety. But again, anaerobic respiration, it's good enough for small organisms like single-celled organisms, but it does not provide nearly enough energy as aerobic respiration, which is necessary for larger organisms. And this is just a repeat that with respiration or the presence of oxygen, glucose can produce much more energy molecules of ATP, whereas something on the cellular level, like fermentation for single-celled organisms, or in some cases, if our muscles really do need energy and there's no oxygen, you can go through fermentation, but that produces significantly less energy molecules. There are lipid and protein catabolism, where protein, through extracellular proteases, break down proteins into smaller components of amino acids, and deamination, decarboxylation, dehydrogenation, and desulfuration essentially break them down and convert them into organic acids, which can be used in the Krebs cycle. So if you have fats or lipids, a lipase can convert it into a different fatty acid, and beta oxidation can convert it into acetyl coenzyme A. which is a starting point for the Krebs cycle. And also, it can break down that lipid into its other component. I should say that lipids consist of a glycerol and a fatty acid. So, essentially, breaking those two apart, both components, the fatty acid and the glycerol, can eventually be converted into acetylcoenzyme A to be used to cause the Krebs cycle to occur. Now, as far as when various organic molecules are used, essentially, while glucose is the typical starting point for how we produce our energy, fats and proteins, if necessary, can be used to get to certain parts of glycolysis, the Krebs cycle, and produce energy. Carbohydrates are more effective as a starting point, but if necessary, our bodies know how to use in a dire strait, how to use proteins and lipids as well. Biochemical tests are useful for identifying bacteria by detecting enzymes unique to that bacteria, as well as those involved in dicarboxylation and dehydrogenation. Just an example of amino acid catabolizing enzymes in a lab situation. Now, fermentation test is where bacteria that catabolize carbohydrate or proteins then produce acid, causing the pH of the solution to drop down to the point where an indicator will change color, indicating the presence of that bacteria. Also, there is the oxidase test, which identifies certain bacteria that have a cytochrome oxidase enzyme, such as Pseudomonas. There's various examples of different fermentation tests. Now, photosynthesis. There is the light-dependent, or the reactions of photosynthesis that happens in the presence of light. This involves the conversion of light energy, or this, really what this should say is this involves the utilization of light energy to form chemical energy, such as forming ADP and NADPH. But there are light-independent dark reactions, where ATP and NADPH are used to reduce carbon dioxide into 6-carbon sugar molecules. This process is called carbon fixation, and it's done via the Calvin-Benson cycle. Basically, the main chemical reaction that's occurring during photosynthesis, the oxygenic side, where 6 molecules of carbon dioxide and 12 molecules of water are reacted together through the assistance of light interjecting necessary energy to form a single molecule of glucose sugar, 6 molecules of water, and 6 molecules of oxygen, which we can breathe in. As far as the N-oxygenic side of photosynthesis, 6 molecules of carbon dioxide and 12 molecules of hydrogen sulfide, when reacted together with light energy, can form glucose, 6 molecules of water, and 12 atoms of sulfur. So essentially, this is whether you have a starting molecule of water or a starting molecule of hydrogen sulfide as a hydrogen source for glucose. Pretty fascinating chemistry. And this is just showing a process that occurs in the enzyme complex photosystem 1. It's cyclic photophosphorylation, where essentially an electron gets released from chlorophyll. So essentially, chlorophyll is oxidized, it releases an enzyme or an electron that goes through an electron transport chain, and eventually goes back to chlorophyll, reducing it back to its normal form. The process is started by light energy, and when the electron goes through the electron transport chain, eventually, through multiple processes, ATP is produced, but chlorophyll is used as the final electron acceptor. Now, in the case of non-cyclic phosphorylation during photosynthesis, light can hit photosystem 2 in the photosystem 2 protein complex in chlorophyll, which excites electrons that eventually go through electron transport chain. They enter photosystem 1, and then they once again are released and go through a different electron transport chain to help reduce the coenzyme NADP plus to NADPH. In both cases, in order for electrons to be released from chlorophyll in order to oxidize chlorophyll, light must interact with chlorophyll. This is just a model of the Calvin-Benson cycle, which involves the input of carbon dioxide to eventually produce glucose, as well as other sugars like fructose, maltose, and other things. Key here is that it involves some energy input and some energy, or I should say, it does involve the input of ATP to input energy, and, well, it pretty much consumes energy. Some energy sources, or electron donors, photosynthetic pigments in conjugation with light, or glucose combines with glucose, elemental sulfur, ammonium, or hydrogen gas. Essentially, electrons come from these sources. And in the process of moving through electron carriers, helps produce ATP to form NADPH or NAD plus NFAD, which then further carry the electron further down the chain to form more ATP, or eventually the electron is accepted by some final acceptor. It could be oxygen in the case of aerobic respiration. It could be molecules such as nitrates and sulfates in anaerobic respiration, or in the case of fermentation, it's going to be some organic molecule. And this is just a large display of how organisms take an initial energy source and either through chemical means or light energy means convert it down the way to form other molecules and eventually transport electrons down the chain to produce ATP. What's common throughout is that you need some sort of carbon source that basically is involved in the process of converting from one molecule to another. There's a wide range of possibilities that organisms have available, depending on the type of organism. Phototrophs, they use light energy. Essentially, it hits chlorophyll, the chlorophyll becomes oxidized, and the electron goes down the electron transport chain, reducing ADP and inorganic phosphate into ATP, or I should say converting ADP and inorganic phosphate to adenosine triphosphate. There are photoautotrophs, which use energy in the Calvin-Benson cycle to fix carbon dioxide or to combine carbon dioxide into sugar. If it's an oxygenic process, it produces oxygen in the end, and if it's anoxygenic, it does not produce oxygen in the end. As animals, we prefer when plants go through an oxygenic process to produce oxygen. Now, Photoheterotrophs, they can use organic compounds as sources of carbon, and therefore they can produce things in anoxygenic ways. These can be actually many unicellular organisms as well. Now, chemoautotrophs, they use energy from inorganic chemicals such as carbon dioxide as well as a carbon source. And energy is used in the Calvin-Benson cycle to basically combine or fixate carbon dioxide. Now, the use of energy and carbon from organic chemicals, essentially glucose in the process of being broken down into two pyruvic acid molecules. In the same process, NAD plus is converted to NADPH, which can then be converted back into NAD plus by becoming oxidized, and that released electron that is removed from NADPH goes down the electron transport chain to form ATP. Basically, this is a medically and economically important process because it produces a large amount of ATP for every 1 glucose. So, basically, organisms use different pathways. Another molecule of importance, instead of adenosine triphosphate, is uracil triphosphate, or UTP. But this is just involved in typically some processes in animals and bacteria. And what we're basically seeing is the combination of glucose to form glycogen, which is where glucose is stored for later. Now, as far as the biosynthesis or the natural production of simple fats and lipids, well, essentially in the glycolysis process, there is an enzyme or there's a molecule glyceraldehyde 3-phosphate, which can be converted to dihydroxyacetone phosphate, and when acting on a glycerol, can form a simple lipid when combining with a fatty acid component produced along with acetyl coenzyme A. The pentose phosphate pathway, which involves pentose or 5-carbon sugars, or the acetyl coenzyme A. Krebs cycle pathway, which can lead to the amination or transamination, where basically the R group of an amino acid is moved from one amino acid to another, or the Tortora pathway, are all possible ways of actually producing amino acids through amino acid biosynthesis. Transamination, again, every amino acid has a lot of the same components, but there's an R group that's very unique. And the R group is what differs one amino acid from the other, such as what is present in glutamic acid and oxaloacetate acetic acid. Transamination allows the movement or the transfer of certain parts, which eventually forms alpha ketogluteric acid and aspartic acid, where aspartic acid is a different amino acid from glutamic acid, essentially 1 carbon unit less. The biosynthesis of purines and proteins and nucleotides, well, essentially forming the other macromolecule, which would be nucleotides, used eventually in DNA and RNA. Basically, your body, the processes that are used to produce energy, also can help produce a wide range of other substances that are necessary in the body. Now, as far as integration and metabolism, there's amphibolic pathways, which are basically metabolic pathways that have both anabolism and catabolism. Anabolism, the buildup of larger molecules, as well as catabolism, the breakdown of large molecules. Many pathways function simultaneously and have common intermediates as well, actually. That material is more appropriate if you take a separate biochemistry course or a cellular and molecular biology course. And thankfully, that concludes Chapter 5 of this lesson of Module 2. There will be one more, thankfully, short lesson about differences in prokaryotic and eukaryotic cells.